Howdy, folks. Thanks for checking in to Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. In this lively notes cast, we're going to be talking about ATP. And we're going to use that as the springboard to talk about respiration and metabolism, even photosynthesis for that matter. The point, of course, of respiration is to make ATP. And we're going to be talking about the ins and outs of what ATP is and how it can function and what it's for. Let's take a moment to talk a little bit about how life seems to require energy, especially in the morning. And really, if you think about it, caffeine isn't really, coffee doesn't have a whole lot of calories, so it doesn't really give you a whole lot of energy. It turns out that caffeine actually just allows your cells to tap into energy stores that they already have. And that's what this video is all about. How do cells tap into their energy stores and use them? Back when we were talking about enzymes and Gibbs-free energy, we talked about endergonic and exergonic reactions. And you may remember talking in chemistry about endothermic reactions and exothermic reactions. Exothermic reactions give off heat. Endothermic reactions require heat. Well, those are versions of endergonic and exergonic reactions. Endergonic reactions require energy. We're just being more general. We're not specifying what kind of energy, if it's heat energy. Or exergonic reactions, which give off energy, which transfer energy to something else out of it. So what do we need this energy for? Well, we need to build things from smaller molecules so that we can assimilate them and make them more like us. We need to organize DNA and get them to move around the cell in the right way, and that means we have to move. Heck, we got to move around like us big multicellular things, and even things with just cilia and flagella, waving them around, and just to move things across the membrane, which are too big to fit or are too polar to fit or are going against the concentration gradient, all that stuff, and quite a bit of us homeotherms, quite a bit of our caloric intake goes into just staying warm, keeping our enzymes at their optimal range. If it's not my morning coffee that's giving me the get up and go in the morning that I need, then what is the source of this energy? The energy that's required for all of those endergonic reactions of life. Well, we do this by coupling those endergonic reactions with exergonic reactions. So those exergonic reactions are catabolic reactions, things like digestion, that when they break apart something, the energy that used to be tied up, the potential that was tied up in those molecules can go into something else. And in this case, and we're not talking about heat now, so we're not talking about exothermic reactions, although sometimes breaking these molecules down is exothermic, we just call that combustion. In this case, the energy is transferred to an endergonic reaction, a synthetic or anabolic reaction of life. Just like there are theoretical crossovers between economics and ecology, we can use an economic lens to look at the economics of a body system and how we have to consume real high quality, high calorie foods, delicious carbohydrates and lipids and proteins and even nucleic acids. Our body's going to break them down. It's the job of our digestive system to bring them into a form that can be absorbed. And we, on a cellular level, we use this catabolism, this breakdown of these molecules, to transfer that energy from the food molecules into a form that can be used by the cell. And that's really the job of our digestive system, like I said, to bring that food close enough to us, give it a big old hug, and break it down so that it's in a form that can be absorbed. And just like in economics, there needs to be a currency, something that can transfer value from one place, from one individual to another. In the body systems, in between from one cell to another, and even within the cell, there needs to be some sort of currency, some way to pass around this energy. We need a short-term store of an energy molecule that can be used within the cell, and that molecule is ATP. Good old ATP, adenosine triphosphate. And if you're thinking to yourself, wait a minute, didn't we talk about adenine back in DNA and RNA? You did. if I take an adenine and I add a ribose or deoxyribose sugar to it, I get adenosine. And if I go ahead then and stick a phosphate on there, I get adenosine monophosphate. That little I there next to the P, that's inorganic phosphate. It doesn't have any carbon or hydrogen in it. If I take another inorganic phosphate group and I stick it on there, I get ADP, or adenosine diphosphate. If I go ahead and stick another phosphate on there, I get ATP, adenosine triphosphate. And it is a synthetic reaction, and so it does take some energy to make the bonds and put them together. And actually, it takes a little bit, maybe a little bit more energy to put those together, because those phosphate groups don't really want to be together, and so it's a little unstable. And so because it takes a little bit more of an investment of energy to make those bonds, we sometimes call them high energy bonds. So that's a little bit of a misnomer, because they're just like any other of those covalent bonds. Let's dig a little bit deeper into those high energy bonds between the AMP and adding the phosphate group to make ADP, and adding the third phosphate group to make ATP. All right, each of those phosphate groups, remember, is quite negative, and so it's a little bit harder to add each phosphate group onto the end. That means once we get into ATP, there's quite a bit of stored energy. You think of it as a lot more potential energy, because you had to put that much more energy into keeping all those phosphate groups on there. In fact, the third phosphate group is the most unstable, and so it has the most stored energy because it was the hardest to stick on there. Those negative, electronegative oxygens with all of their paired unbonded electrons around them and all that electronegativity, putting all those all those electronegative things together makes them repel. And so that molecule, the ATP molecule, is actually pretty unstable. You can almost think of it as spring-loaded, and that P will pop off, and actually the second P will even pop off and release the energy. And this instability actually makes it a really good candidate for something that will hold energy, but also something that will give it away, make it a good energy donor. If ATP is the currency, then how does that energy get transferred from one reaction to another reaction? We're going to take that ATP, and we're going to break one of the phosphates off and turn the ATP into ADP. Now, the amount of energy that used to be holding that phosphate group on, we're going to say is released. Now, I can't take energy and pour it out and hand it to you. All right. And so we're going to talk about being released, but it's not like going away. It's being used to do something else. So just hold that thought for a second. All right. And the energy that is involved in holding that phosphate on, which is no longer holding that phosphate on anymore, is 7.3 kilocalories per mole. All right. So that's our change in free energy. We're going to use that change in free energy to actually fuel other reactions. And how we do this is by transferring that phosphate from one group to another through this process called phosphorylation. And what ends up happening is this phosphate group is kind of like a bad boyfriend. It doesn't really bond very well, and it destabilizes the molecule that it's bonded to. And so when it bonds to the ADP molecule, making it ATP, it makes it kind of unstable, and it doesn't really, it doesn't stay bonded very long, and it breaks off and goes on to another molecule. I can take that phosphate and add it somewhere else, and I can destabilize that molecule. And it's really this destabilization that is the energy that ATP is giving. So it's less energy like a fuel, like something is going to directly that you're going to directly burn, and it's more chemical potential, that instability that it then means when it's unstable, it's going to then go through a chemical reaction, and that's what we need, right? That's what life is, a chemical reaction. These enzymes that drive these phosphorylation reactions from one molecule to another are called kinases. And think of kine like energy, like kinetic energy. Of course, in this case, we're talking about potential energy, but that's the idea. An enzyme that phosphorylates is called a kinase. Polymerization is going to provide us with a pretty good example of how phosphorylation is going to work to drive a synthetic pathway. Here's the dehydration synthesis of maltose. It takes as an endergonic reaction 4.2 kilocalories per mole to destabilize those glucose monomers. They're pretty stable by themselves, and so we have to add energy to destabilize them so that then they'll react. We do this through phosphorylation. Take the ATP molecule, and the phosphate group from the ATP molecule is transferred to the glucose, and that's going to, if you can think of it as restabilize the ATP into ADP and destabilize the glucose from glucose into glucose phosphate. Now, if I take that, that's going to give us 7.3 kilocalories per mole, which is more than enough destabilization to drive the chemical reaction. So the kinase enzyme has moved this phosphate group over. If I hold this destabilized glucose phosphate next to another glucose molecule, I can cause the formation of maltose. I get a little phosphate, of course, and the difference, that 3.1 kilocalories per mole, is the chemical potential that is now locked up in that maltose molecule, so that when the maltose breaks apart, there's that chemical potential that can be transferred to some other chemical reaction. In another example, it's obviously a little more complex, here is going to be the first steps of cellular respiration in glycolysis. Glycolysis is the breakdown of glucose to form ATP, not from the glucose, but to phosphorylate ATP. It actually ends up making pyruvate, but we'll talk more about that later on. The important thing is that we start out with glucose, which is a 6-carbon sugar, and I've written it out here in straight chain. It is in a ring, but I have it in chain form here just to help see how it changes. And through the action of hexokinase and phosphofructokinase, these two kinases, and I hope phosphofructokinase sounds familiar because we did talk about that during our discussion of enzyme control. But those two kinases are going to take the ATPs and dephosphorylate them into ADPs and use those phosphates to stick onto either end of the glucose molecule, and it forms fructose 1,6-bisphosphate. It is in ring form, like I said. And then that fructose 1,6-bisphosphate is actually cleaved in half to form dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. It doesn't matter that those are going to end up going on down the pathway. Actually, the dihydroxyacetone phosphate becomes converted into glyceraldehyde 3-phosphate, but whatever. Those are going to continue on down the pathway and have some more ATP wrung out of them. But these first ATP molecules donating their phosphates to the glucose forming fructose 1,6-bisphosphate is what is going to destabilize that, is what's going to provide the activation energy necessary to actually allow the breakdown of the glucose into the two 3-carbon molecules. With all of this phosphorylation, taking the phosphate group off of the ATP, we're going to need a source of more ATP because we can't store ATP. It's great at transferring that energy, that destabilizing energy, negative 7.3 kilocalories per mole, but it's not good for long-term storage because it's way too reactive. That phosphate group it's too unstable. It's going to phosphorylate. It's going to transfer. So it's really only good for quick short-term energy. If we want longer-term storage, we're going to have to take that energy from the ATP and use it to drive those polymerization reactions and make maltose and then amylose and make even larger fats and make other energy storage molecules. So we need some way to make more ATP from the ADP itself with that phosphate group. And that's where cellular respiration is going to come in, and it's got a lot of work to do because the amount of ATP on a molecule basis is pretty enormous. That starts to bring into perspective the awesome responsibility of ATP synthase which is the enzyme that is chiefly responsible for making most of the ATP for us aerobes anyway. It is an enzyme that's a transmembrane channel enzyme in the inner membrane of the mitochondrion. Remember, the mitochondrion has a big bag stuffed inside of a smaller bag. And in the space in between those two, there is a buildup of hydrogen ions. So there's a whole bunch, much more acidic, there's a whole bunch of hydrogen ions there in between the inner and outer membrane. And because this ATP synthase is permeable to those ions, they flow down the concentration gradient. And as they flow down the concentration gradient through this channel, they actually spin the enzyme around. And it's the spinning of the enzyme that actually drives the rephosphorylation of ADP and phosphate back into ATP itself. The question, of course, is how does this proton gradient form in the first place? How do we keep all those protons in that space, especially if they keep flowing back out through this ATP synthase? And that's going to be the subject of the rest of the videos that we talk about when we talk about cellular respiration. But for now, I'm going to hold it for there. Thanks again for checking into Mr. Ulrich's Land of Biology. And if you have any questions, please go to the website and click on the contact and send me some emails, or you can leave messages in the comment section if you're watching this on YouTube. Other than that, I'll see you in class.